We started talking about the eukaryotic cell cycle and mitosis in particular, and what I'd like for you to know about mitosis is that it includes both the mitotic phase, or M phase, as well as cytokinesis. And M phase is when the cell is dividing the nucleus and all the genetic material inside the nucleus, and then cytokinesis is the division of the cell's cytoplasm. The rest of the cell cycle is the phase between one M phase and the next is called interphase. And that is where the cell is growing, and then S phase occurs within interphase. And S phase is where the genetic information is going to be synthesized, because S stands for synthesis or DNA synthesis, and it's where the cell spends a considerable amount of time and energy making copies of all of its chromosomes, which carry the cell's genetic information. And so each chromosome is going to contain one DNA molecule that has to be copied completely and accurately in order for the daughter cell to be able to receive a complete copy of the parent cell's genetic material. The two strands of each DNA molecule will need to separate, and then those strands will act as a guide for a new strand once DNA replication is occurring. But once that's done, the nucleus will contain two exact copies of each DNA molecule. So all of the chromosomes will now be paired. And those chromosomes need to be separated and put into two distinct cells that are essentially clones at that point. So those two molecules of DNA are actually attached to each other, and they're not drifting apart. They're not going to move apart from each other. They're attached via a thing called a centromere. And I'll show you an image in a few slides, and that image shows the point where these two chromosomes are attached to each other. And when we look at a replicated chromosome, the centromere is going to be the part of the chromosome that looks constricted, and it's where those two identical molecules of DNA are joined. They're attached to each other. And we would call that entire structure to be one chromosome, but that one chromosome is actually, it consists of two identical DNA molecules that we'll call sister chromatids. And these sister chromatids are what end up being pulled apart and then put into separate cells. It's during the S phase and interphase that the cell is going to make a copy of a protein complex that's called the microtubule organizing center, or in animal cells, the centrosomes. And these microtubules that are a part of the microtubule organizing center will help to organize all of the cell's contents, because they will allow the cell to manipulate and move different substances, for example, the chromosomes, into different poles of the cell, and then facilitate the division of the nucleus. So we can see on the slide that during M phase, the genetic material of the parent cell needs to be distributed to the daughter cell once the nucleus divides and all that genetic information is split into two different cells. And because the goal of mitosis is to distribute that genetic information, one copy of each chromosome, into two daughter cells, it seems that it would make sense for DNA replication to happen before mitosis begins. And so what we see is that during M phase, each daughter cell will receive one sister chromatid from each replicated chromosome. And if you can remember, we said that human cells contain 23 pairs of chromosomes, or 46 individual chromosomes in all. So mitosis involves four basic stages: prophase, metaphase, anaphase, and telophase. And then there's the last phase, which is called cytokinesis, which involves the physical splitting of the cytoplasm. And at that point, we have two distinct cells, two cloned cells. All right, so let's start with prophase.
Cells can form many shapes, depending on their function. A common feature of cells entering mitosis is the rounding up of the cell into a sphere. Centrosome organelles and their microtubule fibers then migrate around the nucleus to the opposite poles of the cell.
All right, so during prophase, that's the first phase of mitosis, there were many things that happen. First, the chromatin, which is the loose complex of DNA, need to condense. And the DNA, remember, is carrying that genetic information in the form of a code that eventually gets converted into RNA and then translated into protein. And once those chromosomes have been replicated, which has already happened by this point, the chromosomes are really not distinguishable because they are so spread out and they take up all of the nucleus, the space in the nucleus. So the goal of prophase, or the focus of prophase, is to condense the DNA so that it can be moved around the nucleus and then throughout the cell a little bit more easily. And so it's during prophase that those chromosomes gradually become visible under a light microscope. And as you can see from the slide, the different chromosomes are shown in purple, and they are condensed so that they don't end up getting tangled or broken. And so once they're condensed, then the nuclear membrane breaks down, and the lipids and proteins that make up that nuclear membrane will still be physically present in the cytoplasm, but they're no longer going to serve as a barrier that stops the chromosomes from moving around the entire cell. And so the nuclear membrane has to be dissolved in some way, and then the microtubule organizing centers, or the centrosomes, which are near the nucleus, they end up moving apart from each other, and they separate and go towards the poles of the cell. And as prophase continues, those centrosomes will eventually have microtubules that grow from that centrosome, and they lengthen and they basically attach themselves to the chromosomes. And the point of contact, the place where the microtubules attach to the chromosomes, is called a kinetochore, and there will be one kinetochore that is on each of the sister chromatids, and essentially these microtubules will be able to pull those chromosomes to opposite ends of the cell once we're ready to move those chromosomes. But prophase is essentially DNA condensing, spindle fibers forming, and those fibers attaching to the kinetochores of the chromosomes.
During prophase, chromosomes are tugged back and forth towards the mid-region of the cell.
Okay, so during metaphase, once those spindle fibers have been attached to a kinetochore, they're going to pull the chromosomes towards the microtubule organizing center. But as soon as the chromosomes begin moving to one pole, the spindle fiber attached to the kinetochore on the other sister chromatid is pulling the chromosome toward the microtubule center at the opposite pole. And so it essentially sets us up for a tug of war where the chromosomes are being pulled in opposite directions. And that is a good thing, because that allows us to move those chromosomes into the middle of the cell, where they are essentially equidistant from both of the poles. And we call that situation the metaphase plate. because it is where all of the chromosomes are aligned in the center, and it's at this point where the cell can ensure that the chromosomes are properly replicated before we eventually divide the genetic information into two separate cells. Okay, so during anaphase, this is the next stage of the cell cycle. These sister chromatids are tightly paired to each other because they not only are connected via the centromere, but there are also other proteins called cohesin proteins that are holding the DNA molecules together along their length. And at the start of anaphase, these cohesin proteins need to be broken down, and the sister chromatids will then be able to separate once that protein is broken down. But because microtubules are already attached to each kinetochore and they're pulling, the separated chromatids will be dragged towards opposite poles of the cell, and we will see that the chromosomes take on this V-like shape as they are pulled to the two sides of the cell. And so we can see the two sets of chromosomes being pulled to the opposite poles in the image here.
At the start of anaphase, the cohesin protein that holds pairs of chromatids together breaks down. The separated chromatids then migrate to the opposite poles of the cell.
Okay, and then the last phase before cytokinesis is telophase. And telophase physically it means end, or the end of the cycle, and so this is where the two sets of chromosomes are approaching the cell's poles, and we prepare for the membrane to divide physically. Microtubules are not needed at this point, and so they begin to disintegrate, and the bits of membrane material that's in the cytoplasm can reform the nuclear membrane around the chromosomes that have just been separated. And so the chromosomes themselves, which are each a single DNA molecule, end up becoming less condensed. They open up, and as a result of them opening up again and spreading out, they become more accessible in order to be read as the instructions that the cell needs to know what to do when. And essentially, by the end of telophase, individual chromosomes will no longer be visible, and the cell will be ready to divide into two distinct cells. And so the process of us dividing the cytoplasm is occurring via cytokinesis. And so we can think of the steps of mitosis as being the steps where the contents of the nucleus get divided into daughter cells. It's through cytokinesis, which sometimes happens actually during anaphase or telophase, that the cell's membranes will divide, the cytoplasm will be divided. And the way that this happens is through the formation of a ring of microfilaments. And microfilaments are basically thin strands of protein, of actin protein, which give the cell its shape. And you can think of these microfilaments as like a drawstring bag, where the microfilament ring will pinch the cell in half. And each daughter cell, as a result, will receive a portion of the parent cell's plasma membrane and cytoplasm, along with all of the organelles, like the mitochondria, the ER, the lysosomes, the peroxisomes, the chloroplasts, and so forth and so on. And we will be able to form two separate daughter cells that can both function on their own. Some cells don't undergo cytokinesis, like muscle cells, for example, do complete mitosis, but they don't do cytokinesis, meaning the cells contain multiple nuclei inside one cytoplasm. And those are called multinucleated cells. In terms of plant cells, because plant cells are surrounded by a rigid cell wall, those plant cells can't pinch in half. So instead, a thing called a cell plate will form midway between the two daughter nuclei, and then vesicles from the cytoplasm will eventually deliver cell wall material to the cell plate, where it will physically reconstruct a new cell wall between the daughter cells. And so those vesicles would contain lipids, they would contain proteins that allow the cell wall to be reformed from a brand new set of raw material. The pinching does not happen in plant cells the same way it happens in animal cells. But the end result